Greetings everyone! The topic of our video today pertains to a groundbreaking discovery in neuroscience that sheds light on how the brain overcomes fear. Researchers at the Sainsbury Wellcome Centre at University College London have identified the precise brain mechanisms that allow animals to suppress instinctive fear responses, a finding that could revolutionize treatments for fear-related disorders such as phobias, anxiety and PTSD. Without further ado, let us delve into our discussion. In a study published today in Science, Dr. Sara Medeiros and Professor Sonia Hofer led a team that mapped how the brain learns to disregard perceived threats that turn out to be harmless. Humans, much like animals, are born with innate fear reactions. Think of a child startled by a loud noise or a fast-approaching object. Yet through experience, these instinctive responses can be overridden. For instance, children often learn to enjoy fireworks despite their initial fear of the loud explosions. The researchers sought to uncover the neural processes behind this form of learning. To explore this, the team designed an innovative experiment using mice. They exposed the animals to an overhead expanding shadow, simulating an approaching aerial predator. Initially, the mice fled to safety, but with repeated exposure and no actual danger, they learned to remain calm. This behavioral shift provided a model for studying how fear responses are suppressed. The researchers focused on a brain region called the ventrolateral geniculate nucleus, known to inhibit fear reactions when active. The ventrolateral geniculate nucleus also receives significant input from visual areas in the cerebral cortex, prompting the team to investigate whether this neural pathway plays a role in learning to disregard visual threats. Their findings revealed two critical components. Specific regions of the visual cortex are essential for the learning process, and the ventrolateral geniculate nucleus stores the memories formed during this learning. Dr. Medeiros explained, when we inactivated certain cortical visual areas, the animals could not learn to suppress their fear responses. However, once the learning had occurred, the cerebral cortex was no longer necessary. This challenges the long-held belief that the cerebral cortex is the primary hub for learning and memory. Instead, the subcortical ventrolateral geniculate nucleus appears to store these crucial memories, bridging cognitive processes in the neocortex and instinctive behaviors mediated by the brainstem. The study also uncovered the cellular and molecular mechanisms behind this process. Learning to suppress fear involves increased neural activity in specific ventrolateral geniculate nucleus neurons, driven by the release of endocannabinoids, molecules known to regulate mood and memory. This release reduces inhibitory input to ventrolateral geniculate nucleus neurons, enhancing their activity when the visual threat is encountered and ultimately suppressing fear responses. The implications of this discovery are profound. Professor Hofer noted, our findings could help us understand what goes wrong in the brain when fear regulation fails, as seen in phobias, anxiety, and PTSD. While instinctive fear of predators may be less relevant to modern humans, this brain pathway exists in humans too. Targeting ventrolateral geniculate nucleus circuits or localized endocannabinoid systems could open new avenues for treating fear disorders. Looking ahead, the research team plans to collaborate with clinical scientists to study these brain circuits in humans, with the ultimate goal of developing targeted therapies for maladaptive fear responses and anxiety disorders. To support our channel's growth and ensure broader awareness, kindly hit the like, share and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.